Hi, everyone. Dr. Wiseman here. Welcome to Chapter 3 of APHY 101. I've been meaning to do these lectures in video format for a couple of reasons. One is because the class moves so fast and we have, relatively little time, it's nice to have a backup. So if for some reason you're not catching the material, I really don't want you to dictate. I want you to understand. So I'm going to try to give this in a relaxed way that will allow you some opportunity to maybe go back, hear it again, that sort of a thing. Yeah, the selfie here. So we're doing a cell lecture. You know, many of us are going to be in careers that are, shall we say, macro. That is, you're going to be working with a whole patient, and it may feel like cells are maybe not as important or specific to your career aspirations as perhaps other things. And that's not untrue in terms of, your day-to-day stuff, but all of the therapies, all of them involve cells. So it's a good idea. The more you know, the stronger you'll be. And this goes in every direction, whether you're a parent or a spouse or a loved one or a caregiver in some way. We're all dealing at the cellular level, with things like nutrition and so forth. So let's get going. Okay, so this is something that most of us already know. That is the idea that the cells are, shall we say, building blocks. That's an interesting analogy, a little bit static, but nevertheless, it's part of the dogma of biological understanding that cells are the smallest life form. Yes, we have things that are smaller than cells, but they are subcellular, or they are things that take advantage of cells. Like, for instance, viruses are not living things. They have genetic material and they have proteins, sometimes even some carbohydrates in there, but they can't do it without cellular help. They can't propagate themselves. So cells are the smallest living thing we know of. According to this slide, we have about 10 to 15 trillion cells. That's with 9 zeros behind it. It's probably higher than that. I would even guess double that. But for testing purposes and that sort of a thing, that's the number we're going to roll with, at least for now. But have some awareness that it's probably higher. And we turn over cells every day, so it's a pretty amazing process. There are, as we currently know, somewhere around 250 different cell types. That's amazing. That's because a liver cell is different from a skin cell, which is different from a neuron, which is different from a pancreatic cell, and there are different pancreatic cells. So it's pretty amazing stuff. Lots and lots of different cells arise from the same genetic material that we all inherit from our parents. Now, that previous slide was meant to be what we call a wow slide. Oh, we can get you all excited. As biology professors and instructors, we want you to be excited about this stuff. So let me give you a real wow. This is a picture here of a single cell. You can almost catch the margins of it. What we have done is we have introduced what's called a green fluorescent protein. Originally comes from a jellyfish. We modified this cell so that these green fluorescent proteins are located in a very, very specific place in the cell. And I want you to watch this video real quick. This was taken at 1 frame per second. These things are moving along microtubules, and we're going to hit it with an inhibitor here for a second. Boom, like that. And they stopped. That was really wild. But then even with the inhibitor, they got going again. That was the thing we published this on. But it was really, really exciting. And you can see the dynamics in that cell. You can see how dynamic even just a relatively static looking cell. Here's the nucleus right here. Okay, that's why it's a little darker there, because there isn't any of that green protein inside the nucleus. So you can see just how amazing these cells can be, and we want you to be excited, because when you are geeking out on this stuff, you're learning, and that's what we want you to do. So a little bit of an aside, but I thought you'd find that interesting. Wow is what I would say. And as different And amazing as cells are, there are some things that all cells have in common. Yes, they are the smallest known unit of life. Like I said, there are smaller things, they just are not living. They don't have all of the capabilities and functions of something that we define as living. There's about 7 or 8 criteria, maybe 9. In any case, cells can accomplish those different tasks. It's the smallest unit that can. All organisms, all life that we know of is made up of cells. Now, we are on the verge of some really exciting technology going out into space, going to places where we think life might exist. That being said, maybe we will discover something new in these places. But for now, the only life we know of for sure is here on Earth, and all of the life on Earth is made up of cells. As far as we know, all cells come from pre-existing cells through cell division. And there was a lot of controversy over the last several thousand years where people have been thinking about these things. That life could originate, like for instance, it used to be believed that maggots, which are the embryonic structure of a fly, they turn into a fly, that maggots arose from dead meat, from rotting meat. Well, that wasn't true. The flies would land, lay their eggs, and then the eggs would have maggots on them. That's because the maggots, the cells in those maggots, arose from the cells in the fly eggs. So all cells we know of arise from pre-existing cells through cell division, mitosis, meiosis. We're going to get into that in about 30 slides or so. Lastly, when you see the word cyto, that's a prefix, and you see what's called a suffix at the end of the word, like lymphocyte, we are talking about a cell or a cellular structure of some kind. Cytoplasm, for instance, that's the gelatinous-like material inside cells and so forth. Cytokinesis, the splitting of cells. So anytime you see those things, you're thinking something about the cell theory here. As different as organisms are, and we have kind of two major divisions here, we're going to break this out a little bit more. As different as cells in our world are, they all cells, assuming we have this cell theory, all cells have these things in common. They all have cell membranes. Now, eukaryotes also have what are called organelles, which are membrane enclosed structures. Part of the reason why eukaryotic cells can be so much bigger than prokaryotic cells, bacterial cells, is because of that organization. They are much more efficient metabolically because they have organelle membranes. Prokaryotes don't have that luxury, but they do have a cell membrane, and that's where most of the metabolic activity occurs, on the inside of that cell membrane. All living things that we know of have DNA as their core genetic material in the way that they transfer information about the organism from generation to generation. There's no exception to that. Now, we do see viruses that are RNA, and we see some other things like that. Even like the HIV virus, which is an RNA virus, turns its RNA back into DNA once it infects a cell. The third thing that all cells have, regardless of where you find them, they all have this structure. And you could say it's this protein, but it's actually a mixture of things. It's got protein, it's got RNA, it's got some other things. All cells have ribosomes because that is the structure which converts the language of a nucleic acid, that nucleotide sequence, into the language of an amino acid sequence, which then gives rise to proteins. And what's interesting about ribosomes, just for a second, ribosomes don't change that much. Bacterial ribosomes are really similar to eukaryotic ribosomes, not identical. In fact, there are some really exciting antibiotics out there that can exploit the differences between prokaryotic bacterial ribosomes and eukaryotic ribosomes. And so stay tuned. There's a lot that's going to arise just from this very simple information. Now, I did mention this in the previous slide that eukaryotes have organelles. So animal cells, mammalian, reptilian, avian, fish, and so forth, all have a specific nucleus. All right. You can see one here in a plant cell. It's a eukaryote as well. And they have membranous organelles. You can see mitochondria in here, got chloroplasts, and some other things in here. Fungi, for instance, you think of mushrooms or yeast, like for fermentation products and things. They also have membranous organelles. That's one of the reasons why fungal infections can be kind of hard to treat, because they're very similar to our animal cells. And so if you give a poison to them, it's likely it could be poisonous to our own cells. That's why we run a little conundrum there on some of these things. The last one here is called a protist. And now I'm not going to break out protists too much, but protists are kind of weird. They kind of share characteristics of both animals and plants, but they are eukaryotes. They have nuclei. Some have two. It's kind of crazy. They have nuclei and they have membranous organelles, and that's really all we need to focus on here. They are eukaryotic because they have a dedicated nucleus and they have membranous organelles. And so we have some pictures here on the left. This is a stained cell. A cell normally would not look like this. This is under a fluorescent microscope where they have given different markers in order to mark different structures inside the cell. The nucleus is blue, and we've got some membrane structure, which is green, and some orange in there. But it doesn't really look like that. It's just that when we stain it, it makes it easier for us to see some of this stuff. You should start to familiarize and get yourself maybe a 4 or 5 word explanation for each of these things. A little haiku, if you will. Okay? A little haiku, a little short little phrase, something that will remind you of what you actually know about each of these different things. Okay? So, yes, we are in anatomy and physiology course, and this includes cellular anatomy. So, since we're not dealing with cellular anatomy so much in pure anatomy, cytoanatomy, if you will, because we're physiologists, we have to cross that line a little bit, and this is one that you're going to want to have to cross. So get started with these. We are going to break out the functions of some of these, and as always, if you have any questions, just hit me up. I'll be glad to answer those. So we're going to start way down in the nucleus, one of the most interior compartments of the cell, talking about these structures called chromosomes, and within those chromosomes, something called genes. If you look in that previous picture of the cell on the left, it doesn't look like this. It's more homogeneous blue all the way across that nucleotide. That's because this chromosome structure, which is an individual segment of DNA. All right, I've got some text going to come up here in just a second. This picture of this chromosome right now, there are two things about it that we need to know. One, this is like a packed suitcase. All right, you go on a trip, you pack your suitcase, you get to where you're going, you can't just have a packed suitcase. You've got to unpack it in order to get access to the clothes and your toothbrush and et cetera, et cetera. This is a packed suitcase. So it's packed up. Normally it isn't. So if you look at the homogeneous blue on the previous slide on that cell on the left, that's an unpacked suitcase. Secondly, this chromosome is about to be split in two. This is a pre-mitotic chromosome. So there's one chromatid on this side, another chromatid on this side. They're going to split. And so one copy is going to go to one daughter cell and the other copy is going to go to the other. So there's a little couple of things. I don't want you to misunderstand when we're showing you this picture that it's always like this, and here it isn't. All right. Now inside all of these chromosomes, and you have 46 total. There are 44 autosomes. All right. Talk about autosomal recessive disorders. Okay, cystic fibrosis, for instance. There are 22 pairs, 44 in total, of autosomes. These are what we call non-sex chromosomes. They generally don't give rise to sexual characteristics. Then you have a pair of sex chromosomes. In most individuals, we have either an X or double X, XX for a genetic female, or XY for males. I don't want to wade into the culture war too much, but there are XXY individuals, and there are people that don't fit into those biological niches that we so easily fall into, even if they are XX. Genes over here, and I want to talk about genes in just a second, do jump around on chromosomes. It does happen. Okay, so you have 44 autosomes and 2 sex chromosomes, total of 46, 23 pairs. All right, half coming from mom, half coming from dad. It says 20,000 genes. Is actually a little higher than that, but we'll just leave it at 20,000. So back to the idea of genes. A lot of our DNA, a surprising amount, we don't know what it does. It may have some function that we just have not yet uncovered. But when we have a sequence of DNA in the larger chromosome that gives rise or could give rise to a protein, we call that functional structure a gene. Genes are normally found in very, very discrete and specific locations on chromosomes, but they do have the ability to move around. And I don't want to delve too deep into that, but let's keep with the basics. That genes, the functional unit of DNA that gives rise to a protein, are found inside a chromosome, and chromosomes are found inside the nucleus. So we've gotten the nucleus out of the way. We have other structures in here that you should get to know. And again, the goal here is a simple 4 or 5 word max, 3 words, 4 words, 5 words, a haiku, if you will, of each of these things. Not a recitation, because that's not very productive. All right. Recitation will get you in the door, but it won't get you to function. It won't get you to flexibility. All right. So get to know these things and develop a little phrase for each one. For instance, the nucleolus is where we synthesize ribosomal RNA. So if you look closely at a non-mitotic nucleus, you'll see these little structures in them. They're called nucleoli. What we know about nucleoli is that they are where we synthesize ribosomal RNA, which goes to the ribosome and helps the ribosome be that protein generating machinery. So you have protein in the ribosome, but you also have ribosomal RNA, lowercase r, uppercase RNA. That's all we need to know at this point. Again, we're going to break some of these things out. The key is 3 or 4, maybe 5 words for each of these things, just to remind you of what you already know rather than memorize. Okay? Let's take a look at the plasma membrane. Cell membrane, I don't like that word cell membrane because organelles all have membranes. They're all cell membranes. They're just organelle membrane. Okay, whatever. Okay. The plasma membrane is the exterior surface of the cell most of the time. All right. So we've got plasma membrane structure. And if you remember in the previous slide, I talked about 4 primary constituents. The 4 primary constituents are phospholipids, which are these funny looking little, they almost look like a sperm cell with 2 tails, not one. Those are fatty acid tails. Talked about that in class. We have cholesterol. You can see these little yellowish structures down inside. Cholesterol is very, very important fluidity buffer. Certainly have proteins, and proteins can be on the periphery of these membranes, or they can go through or be inserted in. Lots of different things, okay? We're going to talk about more of that in a little bit. And then lastly, we have this fun word you may or may not have heard of. It's called the glycocalyx. All right. I'll let you in on a little secret. I often do, right? There are very few carbohydrates found inside cells. We find glucose. We find glycogen, which is a polymer of glucose. And then and this is animal cells. I don't want to get into plants all that. Okay, so we've got glucose and we've got glycogen, and then the only other family of carbohydrates we see in any abundance on a regular basis are the ribose sugars, the ones that make up nucleotides. So let's leave that. All the big chunk of the other carbohydrates we find are on the exterior surface of the plasma membrane, the glycocalyx. Why? Because so many of these proteins that we find sticking through the membrane, transmembrane proteins, receptors, and so forth, have carbohydrates on them. We have glycolipids. This is a phospholipid with a carbohydrate chain. We've got glycoproteins. This is a transmembrane protein that has some carbohydrates attached to it, and so forth. Okay, so the glycocalyx is just this region on the outside of the plasma membrane where we see biomolecules linked up with carbohydrates. Right. So again, this is a great picture. It may not be the only picture you see of this. So the key is to not try to memorize it straight on this figure. But again, give yourself some latitude by a little haiku here and there to remind you of what it really is rather than just what this picture is showing you. Membrane proteins are there are a bunch of different functions of membrane proteins, and if I go back to that prokaryote eukaryote argument, if all the metabolic functions are happening on the inside surface of that bacteria, it makes sense that membranes are going to have lots of different kinds of proteins in them, whether you're a prokaryote or a eukaryote. One of the things we'll be most commonly talking about as a function of a membrane in, associated protein, whether it's embedded or if it's just right on the edge of the membrane, is one of a transporter. Now, that's probably going to have to be a transmembrane protein. These will facilitate movement of molecules across a membrane, whether in or out. We also have membrane embedded enzymes. One of my favorites is one called RAS. It's very associated with cancer and so forth. What happens though is that it will facilitate enzymatic activity, facilitating chemical reactions in and around a membrane, whether it's the plasma membrane on the exterior surface of the cell, or we're talking about membranes and organelles like the mitochondria. The mitochondrial membrane is like 75% protein, very relatively little phospholipid. There's some there, but it's mostly protein, and a lot of those proteins are enzymes. We have receptors. Now, what I like to say here is that this protein is transferring information into the cell. It can sense the presence or absence. In this case, it looks like a chemical signal, but it could be a physical one, and in turn causes changes inside the cell which lead to responses to this information. That's why we call it a receptor. All right. Then we have, like, again, glycocalyx. A lot of those glycocalyx structures are proteins and lipids that give rise to individual cell identity. All right. So that the cell knows it's a self or non-self, we're talking about immune, or that the cell is properly located, if we're talking about just any old cell, it needs to be in the right place, and this helps with that, the cell surface identity markers. We also have proteins, and we'll talk about junctions in a little bit, that assist with cells sticking together or adhering to other things like connective other tissues or to fixed points. Like for instance, a muscle cell has to have adhesions to the ligaments or sorry, tendons, not ligaments, but that's bone to bone. But a tendon links a muscle to a bone. So you got to have adhesions. The muscle's got to be stuck to the tendon, and the tendon's got to be stuck to the bone, et cetera, et cetera. Then lastly, we have the cells often have to have a discrete structure, all right? And that structure involves what's called the cytoskeleton. Like think of a red blood cell. It's kind of a biconcave. It's got a skinnier middle than the outside. Well, that's due to the cytoskeleton. And in order for the cytoskeleton to do its thing, it's got to be anchored to the cell membrane, whether it's plasma membrane or other things. So here are some examples of cell junctions. I'm going to start with the one in the middle. Super important for physiology, super duper important. You ever hear of cholera? Thousands of people, sometimes millions, have die of cholera. You get cholera outbreaks. San Francisco had a terrible cholera outbreak back 150 years ago. That's because you need to be able to control what is happening passing through membranes, and tight junctions do that. All right, so they are junctions. They call them tight because they're not only holding the cells together, but they're also regulating movement of material between the cells. Okay. Let's go to the one on the right now. This is called a gap junction. This is almost doing the opposite. If we're regulating in this vertical plane with a tight junction through what we call paracellular motion, gap junctions, on the other hand, allow for communication from the cytosol of one cell to another. For instance, let's say this cell right here gets stimulated and it releases calcium. That calcium can not only cause protein function inside the cell here, it can move over here and start influencing protein function in this cell over here. This is literally a cytosol to cytosol conduit. The last one over here is called a desmosome, and there are many flavors of this. Desmosomes are about adherence. First and foremost, there are some communication functions known, but we're just going to say these help the cells stick together laterally. There are some other ones that help the cells stick to basement membrane things, but these are the big three. Tight junctions regulate the movement of material between cells. Okay, gap junctions regulate the movement of material from cell to cell. And then desmosomes are anchoring junctions that help cells stick together. Shifting gears just a little bit, when we talk about transport, you remember in that previous slide when I talked about the tight junction? When we have things moving between the cells, that's called paracellular. When we talk about transport, we're talking about transcellular, moving through cells. All right. And so we have transporter proteins. All right. We got to break this out a little bit. So the two types of transport that we have either require energy, we have to invest energy. That's called active transport. So there's not enough energy in the environment to get that material across the membrane, much less across the cell. Passive transport, on the other hand, means that there is enough energy normally already present that this transport could happen on its own if the membrane allowed it. We're going to break that out, okay? We're going to break that out a little bit. Did you hear how I put in the condition there? There are things that would move if the membrane would let them, all right? If the membrane doesn't let it, yes, the potential is there, but you need to have the membrane allow it. So let's take a look at this. Simple diffusion doesn't need a protein. The molecules are, the word is permeant or permeable. All right, gets a little weird sometimes, but these molecules are generally small, uncharged, and lipophilic. All right. And they can diffuse through that hydrophobic interior. Those are phospholipids there. They can diffuse right through there because they are not impeded by this hydrophobic region. All right. They can move through there just fine. All right. And if there's a gradient, then the energy exists for diffusion to occur. Then we have the other molecules. These things tend to be a little bit bigger than these guys, these molecules here. They are often hydrophilic. They need a protein to facilitate diffusion. That is to say, there's a gradient here. You can see there's more up here than there is down here. So these things would diffuse if they could, and the protein creates the could. The protein allows for diffusion to occur. That's what we mean by facilitated. If it didn't have a protein, it wouldn't be facilitated. It would be simple. These guys right here have a lot in common with the next one. This is active transport. The thing about active transport is that we have to invest energy. Why? Look at the number of molecules up here, down here. The concentration is higher, which means that we have to invest energy because we're pushing more and more in. These guys would actually prefer to go this way from an energetic standpoint. All right. Just like a bicycle would rather roll downhill than uphill, we have to invest energy in that bicycle in order to get it to roll uphill. And in active transport, a lot of the time we are moving things against the concentration gradient, so we have to invest energy. All right. So simple diffusion, no protein. There's enough energy in the environment for these things to diffuse on their own. Facilitated diffusion, we need a protein to assist. There just isn't enough energy. There is the desire, the thermodynamic desire, but these guys can't get through the membrane, so we facilitate that movement using a protein. Always use a protein in active transport, always, 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 and we're always investing energy when it comes to active transport. As I mentioned in the previous slide, diffusion is passive unless it says facilitated. Okay, so when we say just diffusion and there's no prefix on it, we are assuming simple diffusion. There's already enough energy in the environment that this material will move across whatever distance or barrier without the need of assistance. In human systems and in mammalian and animal systems, carbon dioxide, oxygen are small enough, they're generally uncharged, in some cases lipophilic enough to diffuse of their own accord. Small lipophilic molecules like fatty acids, cholesterol, steroids, small lipids, uncharged molecules have an easier time at it. It's sort of depends on the molecule. Many of our drugs can also diffuse on their own as well. Many drugs are lipophilic small molecules. Facilitated diffusion, on the other hand, is diffusion that could occur, but the membrane doesn't allow it. So you need a protein. Either it's going to be a channel or it's going to be a carrier. All right, this one happens to be a carrier, which makes it the most similar to active transport. But this one doesn't require energy, and we are moving these molecules down their concentration gradient. So we are facilitating what would otherwise be diffusion if we didn't have a membrane. We're talking about charged molecules, like for instance, ions move through channels. All right. We have water channels too. We're going to talk about osmosis in a minute, and I'm going to make a clarification here. Large molecules, again, relatively speaking, there are huge molecules out there that we need energy for. But if we're talking about amino acids or small peptides or small biomolecules, nucleotides, et cetera, they need some help. So we're going to use carriers to do that. Move it across a membrane that normally wouldn't allow that to happen. Not up the concentration gradient, it's still going down. Lipid insoluble molecules, again, things that have a charge, things that are aqueous. Glucose is a great example. Glucose doesn't have a charge, but it is water soluble. Amino acids, a lot of them don't have charges, but are nevertheless water soluble. They are being facilitated. Most of the time, when we're talking about diffusion, whether it's facilitated and so forth, we are talking about solute molecules. These are things that are dissolved in the solvent. All right. Biology, the solvent is almost always water. Not a perfect statement here, but it is good for our purposes that in biological systems, the solvent is water. So if the solvent is moving, in this case water, you just saw the animation there a second ago. If the solvent is moving, we call that process osmosis. And what's happening is the solvent is moving down its concentration gradient. Turns out with water and a lot of things that if you have more solute, you have less solvent. All right, I don't get too crazy here. But if the water is moving across a plasma membrane, we call that osmosis. Now, word of warning here. That video that I just showed you, that animation that was that existed in this PowerPoint before I got here, that animation is actually not physiologically relevant. Yes, water can creep through a plasma membrane, albeit slowly, because it's not charged, but it's polar. And polar things don't get through membranes very well. Yes, water can creep through, but most of the water movement that we see in plasma membranes is actually facilitated diffusion, facilitated osmosis. Okay, it's facilitated. We have proteins that allow that water to move. Otherwise, if water could just move on its own, it's so small, we would just desiccate instantly. All right, the water just come right out of our skin, we would be done. So we only allow osmosis when we permit it, which means it's facilitated, not just simple diffusion. Yeah, this can happen, but it's not the real deal. I'm going to take a time out here and call this the end of part one. We've been going a little more than half an hour, and I don't like to make videos longer than half an hour. So I'm going to take a time out right here, finish this video up, upload it to YouTube or wherever I can get it, and we'll pick up with the remaining slides. We're about halfway done in part two. So I'll talk to you soon. Thanks for your time. Bye.